Okay, so exam 2 will hopefully, that was exam 2, right?
Yes.
Will hopefully be returned by next Tuesday. As always, we shall see how long it takes to grade those short answers. And hopefully most of you realize you're only supposed to answer one of them.
Oh, the short answer?
Of the short answer.
Just one?
Yeah.
Yeah, I do too.
Yeah, you guys have to start to read directions. I guess I should read them out loud, but I feel like you guys are okay with it. But anyways. Okay. So if you answered both, if you have a preference of which one I grade, just email me. Say grade this one or grade this one. Don't tell me now because I'm not going to remember, but email me. Otherwise, I'll probably just go with the first one. Whatever works for you, I'm here for you. Okay. Recitation quiz. Has everyone been able to access the recitation quiz? Okay, so this is just an experiment on my part as a way to avoid having recitation on weeks of exams because the group that has recitation usually is not, it's a waste of their time. So let me know when it's all said and done how you feel about doing this, and if it's good, I'll do it for the third exam too. If it's not good, then we'll have recitation on that same day like normal, but it's just that that group might do really badly on the quiz because they're not going to be focused. So think through it. Email me if you have any thoughts on how it went. So don't forget to take it because you have till the end of the day on Friday. There may be an error. I was just emailed there may be an error. I will let you guys know if there is. Yes. Yeah. So I'm gonna check and see because I thought I did it right, but yeah.
What time is end of day?
End of day is 11:59 p.m. Yeah. Otherwise, I would write COB, close of business. When I worked in biotech, they would always email me like, "Get this to me by COB." I was like, "What the hell is COB?" And I was like, "Close of business." I'm like, "I don't even know what you're talking about." So that's when I knew I wasn't meant to be in biotech. Okay. So we're starting a new unit on gene expression. We'll start with a little discussion to get your brains working. So what is gene expression? It's an easy one. Start easy. We'll get one. What is it? Transcription.
DNA to RNA.
DNA to RNA to protein. Yep.
Production of certain proteins.
Yeah. So the end result is production of certain proteins. Okay. So when does gene expression happen? Okay, so when a transcription factor goes into the nucleus and binds to DNA and says, transcribe this gene. But so that's absolutely right, but let's get a little broader, a little more big picture. Yep.
The environment determines it needs to.
Okay, so when the environment determines that expression of a certain gene needs to happen.
I was just going to say when the cell needs to respond to something.
So when the cell needs to respond to something that needs to happen, yep. Extracellular signals from other cells, right? These are all right? Because sometimes it is just the environment. It's time to go into cell cycle. We need to make cyclins, So that's one thing that's going to happen. That's just kind of like normal process. Sometimes in response to extracellular signals, say growth factors, or sometimes, in the setting of an infection, certain genes are going to be expressed. In the setting of nutrient deprivation, certain genes are going to be expressed. And all sorts of things, right? So the point is I want you guys to understand that this is induced, this is regulated. There are certain genes that are always expressed, but then a lot of genes are only expressed when they're needed. Does anyone have an example they can think of a gene that's always expressed? Hemoglobin, is it always expressed in all cells? But is it always expressed in some cells? Yeah. But do you think it could be inducible also? right? If you have low oxygen, maybe your cells are going to start producing more hemoglobin, right? Give me another one.
RB,
RB. Is it all okay?
cell cycle.
Is RB constitutively expressed or is it inducible? So you're thinking, well, you're always going through cell cycle. Well, are you always going through cell cycle? What's an inhibitor? So you would expect that probably would be expressed at pretty stable levels because it needs to inhibit. I see what you're saying. It needs to inhibit E2F. Is that your logic?
Yeah.
Why not? Someone give me something else that you think might be. Yep.
It's gonna go with skin cells.
So in skin cells, what kind of genes do you think are always expressed?
SCKs.
Okay, so you're relating it to cell cycle. So yep, so CDKs, so you're thinking they're dividing frequently, and so as CDKs are always being expressed? Maybe, and maybe they're just not active because that cycle is not there until it's needed. You might say that it's frequently expressed because they're frequently dividing. What about something that's always expressed at the same level? Yeah. Tumor suppressor genes, like p53. Yeah, maybe. What else? I'm looking for certain things. Yep.
Genes that code for receptors, like gene protein receptors, or
Genes that encode for receptors because they're always there.
I guess maybe they're always there with different levels.
But so then they're going to fluctuate. They're always there, but at different levels. So then you're not always going to have this gene expressed at the same level at the same time. Some receptors might always be there, depending on what they do. Because that's so broad, right? Okay, how about over on this side of the room? A gene that's always being expressed.
I don't know if this makes sense, but what about a gene associated with energy, like ATP?
Okay, so a gene associated with energy, or a gene associated, so you're thinking homeostasis, right? So genes that are associated with maintaining cell homeostasis, like metabolic type genes, maybe? Yep.
Like tRNAs and polymerases.
tRNAs and polymerases, so these are things that are frequently needed. Okay. Okay. What else? You have one that I know you know. Genes that are always expressed. Because you use them as housekeeping genes. That's my PCR. Yeah. Sodium potassium pump. Yeah. Yeah. GAPDH. Beta actin. Ubiquitin. always correct. Yeah. So these are things. So we went from very specific and relating it only to cell cycle, what we just learned. And yes, that's one thing to think about. But then if you go broad and you just think of the normal function of a cell, beta actin, always there, always present. It's very important in just the cell be having its normal shape and its normal function. GAPDH, metabolic enzymes, stuff like that. Proteasome components. Yep. Proteins involved with making ribosomes. Right, so you want to think like, it's nice to think about these things so you can understand the concept of inducible versus constitutive. So certain genes are constitutively expressed, they're always on, they're always being made, cell always needs them. Certain things are inducible, meaning you only need them you only make them when you need them. Okay. So how does a cell know to express a gene? We just touched on that. Something about a transcription factor, right? There's something that makes a transcription factor. So to go back into the concept of RB and E2F, right? Well, how do you know? The whole point of that whole pathway was that you had a growth factor binding to a receptor, and that's going to stimulate activase. That was that whole pathway. And how does that happen? Well, you've got growth factor that's causing the signaling that's resulting in transcription factors going to the nucleus and activating gene expression, essentially. And the ultimate gene that's expressed is S cyclin. and that cycle and then combine to FCDK and activate cells, basically S phase. So it's about getting that signal and allowing a transcription factor to go into the nucleus. The PRBE2F thing was a little bit more complicated than, say, NF-kappa B, where you just have a transcription factor and that's in the cytoplasm that goes into the nucleus and activates gene expression. With E2F, there was multiple levels, right? Because you had to first make MYC. So in a sense, you're upregulating expression of your transcription factor that then will activate expression of your cyclin to then phosphorylate your RB. So it's like multiple levels of regulation. And that's how complicated it can get, or simple, depending on how you see it. Okay. So, and this we kind of just talked about, why would a cell want to express a certain gene, usually in response to the situation. Any questions about the basic big picture concept of gene expression? Because everything we're going to talk about basically for the next few weeks is going to be how gene expression happens, from the function of RNA polymerase all the way to different domains of proteins that bind to DNA and activate gene expression, regulation of gene expression. And so I want you to kind of keep it in your mind of like the whole story, that it's usually kind of in response to something activating expression of a transcription factor that will then go into the nucleus and activate expression of some gene. Okay? Did you have something?
No, I was just gonna say something that like always killed me was like DNA synthesis and transcription translation. I always get like those two kind of confused. So,
Okay, you're not alone. So people always confuse DNA replication and transcription and translation. And I want you to separate them, because that's why I teach DNA synthesis as part of cell cycle. Because that's really what it is. You're copying your genome, giving it to your daughter cells in mitosis. Completely unrelated to gene expression, which is make a copy of your RNA and then turn it and translate it into protein. And so I guess the way to remember is replication, you're replicating. You're copying your DNA so you can give it to your daughter cells. Transcription, you're transcribing. Like, so what is transcribe? Like, if you think outside of science and you think about like a court stenographer, or you think about like a medical transcriptionist, or like these people, what are they doing? What's their job? They're copying information to be used for something, right? Yeah. And often translating from one language into another. Yeah. So exactly. So then you have your transcription, and then that gives you a transcript. Just like if you like your radio show, you'll call up the radio station and say, I really want a transcript of that show. And they'll send you a piece of paper with all the words from that show on it, right? And then you can read it. And it means something to you. Huh? Not anymore, now you just go online. But like in the olden days, before the internet, I don't know what that was like, but people existed then. In the olden days, actually, I grew up during the age of like the beginning of the internet. We had AOL. People went to chat rooms. I don't know, people still do that? Yeah. Okay, I'm digressing. So transcription and translation. So one person once asked me early on in my teaching career, okay, so every time you need to express a gene, you first need to do DNA synthesis, right? No, never. Totally separate process. And I think where it goes wrong is that it's taught together. You're taught replication, transcription, translation, as if that's the order of operations. And it's not, so don't think of it that way. So strike that from your brain. Strike. Okay. questions about this? Does that help at all? I'm here for you. Yes.
General, but I don't remember, what are housekeeping genes?
Okay, what are housekeeping genes? Does anyone know?
Genes that regulate the amount of proteins within the cell and the genes that are not, like it all allow a certain protein to be degraded at a certain amount of time, depending on when the protein is.
Okay, so genes that regulate levels of proteins, I think that's me. That might be one example of possibly one type, because that would be like, that might be one kind of housekeeping gene. But so like, housekeeping genes, who wants to take another stab at explanation here?
Housekeeping gene is pretty much always expressed in the cell.
Right.
Always.
So a gene that's always expressed, and one might be ubiquitin, and it's involved in a process that's always needed. But then there are other things like energetics related genes, cytoskeletal proteins, those things that are always needed are housekeeping genes. Anyone want to add to that?
Activators and silencers.
Huh?
Activators and silencers. That help you to basically increase expression.
So activators and silencers that help you to, so you're saying activators and repressors.
Yes.
Are they housekeeping genes? Not really, right? Because an activator might be upregulated, or a repressor might be downregulated in certain situations. Yes.
This might have nothing to do with this. What about the organelles? Like, are there genes specific for organelle replication, or
Sure.
are they passed on? Like,
Organelles, where do they come from? Is
Yeah.
that your question?
Yeah.
Okay, or are they passed on? Well, so membrane-bound organelles, where do the membranes come from? Does anyone know? What's your major source of a membrane in a cell? Within the cell, yep. Yep, the ER. The ER. So, the ER supplies the membrane for a lot of these organelles. There, I think the genes, so then there are all the different proteins, for example, that work within the different organelles, right?
no, I'm not, like, is there a gene that you need to translate to make an organelle?
No, there's not a gene that you need to translate to make an organelle. Really what's going to happen is the proteins that are going to function in the organelle are going to, so, I feel like I don't have a good way of answering this, but maybe because there's a gap in my knowledge. Yeah. Oh, God, okay. Okay. So he said, what about the mitochondria? That duplicates itself, right? So I don't know. It's not an organelle's gene is not included in the genome. So mitochondria duplicate themselves, and then mitochondrial proteins get made in the cytoplasm and get sent to the mitochondria, for example, and imported in, and they function where they function. ER, that's a very easy one. It's just can expand by controlling the amount of membrane and controlling the levels of ER resident proteins. And those again are your genes for those individual proteins are going to come from your genome and then be translated into the ER and folded and processed in the ER and stay there to work there. Only one big thing. Was everyone satisfied with that? Yep.
I remember you saying that DNA synthesis and transcription and translation are unrelated, but don't you have to transcribe and translate the proteins involved in the DNA synthesis?
Okay. So that, yes. So what Angela just said is, don't you have to transcribe and translate the proteins required to do DNA synthesis? Yes. Yes, because that's how you get all your proteins to do everything in the cell. But in terms of they're not related, what I mean is you don't need to do replication in order for transcription and then translation to happen for each gene. And I think it's because it's kind of taught that way that people believe first you copy your genome, then you make your RNA, and then you make your protein, and that's not the case. I say that like 50 times during this unit because so many people thought that. Like I once took a vote, and I was like, who thinks that? And half the class put their hands up. And I was like, okay, stop. Okay, any other questions before we move into the nitty gritty? Good stuff. No. Was that fun? Okay. Okay, so this picture is really showing the big picture of signal from outside, binding to receptor, signal transduction, something going into the nucleus, and then you have your transcription here. Okay? And the artist's interpretation. Clearly you're missing a lot. But. Okay, so transcription it is. The first step in the process of gene expression. And here, this was taken from a book that propagates in a weird way the misconception, but you have to remember, when you see this DNA, RNA, protein, don't think of it as replication, transcription, translation. Think of it as the information is stored in the DNA, then the cell makes a copy of the gene that's needed, and then that message is translated to make a protein that serves whatever specific cellular function. Okay? So, who was it? Cindy Gent, Dr. Gent. She said something about how it's really nice. You can think of your genome as your cookbook. And your RNA is kind of like, well, I want to make big ziti, so I'm going to look in my cookbook. And then I'm going to, that cookbook's super important, so I'm not going to work directly out of it. Instead, I'm going to copy down my big ziti recipe. And then I'll put my cookbook away. And then that baked ziti recipe is going to be used to make my baked ziti. And so that's kind of, I always like that way of thinking about it. Your cookbook is your DNA, your genome, your RNA is when you've copied down your recipe, and then the ziti is your protein. Okay, yeah?
I'm still having trouble with, I know that we learned that there's like a target binding protein site, like when we were doing replication, the target protein would bind there. But I'm confused as if you have so much DNA and you just need like one specific part, how it knows out of miles and miles of information to go to that one part.
Okay, so Venus is asking, you have a whole genome with miles and miles of information, how does the cell know to go, or how does your RNA polymerase know to go to one site to do transcription? Okay? And so to answer that, you need to think about a few things. One is what we're going to learn about, which is transcription activating proteins and how they work, and how they come and they bind a certain site and activate transcription there through recruitment of RNA polymerase to that location. And then the other thing is you have to merge that thinking with what we learned in terms of chromosome structure and how your chromosome needs to be open there. And so how you're going to get acetylation of those histones and demethylation of that DNA and open things up. But all that's going to be started by, remember, some kind of transcription factor that's going to bind. So if you can imagine, think about what we just talked about with E2F, okay, which activates S-phase gene expression. So E2F is going to go in and it's going to bind to different sites in the genome, right? So let's do, oh, my paper. No paper for me today. Who's got paper? Or I can draw on the board. Sometimes there's a nice big stack there for me. So So let's just draw a quick picture of it, bringing it all together, and you can tell me if this helps you. That's really close. Okay, so Venus's question is, if there's all this genome in your interphase nucleus, how do you end up with one little RNA coming out? Well, it's not really one little RNA, is it? It's many more, but just for simplistic. So, and remember, this is in reality kind of like all super packaged around histones everywhere, right? Okay, so what's going to happen, so that will be our nucleus, and let's make this our cell. And so, you'll have your receptor and your signaling molecule, and all this magic will happen with your signaling. And then you're going to have your, we won't do E2F because I made it signaling. You'll have some transcription. We'll call it NF kappa B, but it doesn't matter. We'll go into the nucleus. It was out here, and now it went in here. Okay, and so then it's going to go in, and it's going to have certain binding sites. So it's going to come in here, and it's going to bind right here. Okay? And so that's going to recruit like all sorts of things. A lot of things are going to start happening. But like one thing will be you'll kind of open up the DNA by modifying your, and this is not specific, let's make it more general than NF kappa B. I don't want to commit myself. Transcription factor. And it's going to recruit those reader-writer complexes and open things up, right? So you'll get acetylation and you'll get demethylation. And so then you'll end up with this big loop of DNA that's open, right? Okay? And so then you'll have other transcription factors, because it's not as simple as there's only one involved. They're going to come and bind here, let's say. Because let's say that's like an enhancer region. And then what it's going to do is it's going to recruit other things. And so then maybe you have some other transcription factors, these general transcription factors that we'll talk about, that will then combine there. And then you get recruitment of RNA polymerase here. Okay? And so now you have it in this certain spot. And so it's going to activate gene expression in that way. And then you'll get expression of this gene. And so you'll get all these RNAs. They're going to be made. And it was all because this signaling sent some transcription factor in and said, let's open up the DNA here. And let's recruit RNA polymerase here. And now we've got a lot of RNA made. And then the RNA, remember, I'm skipping processing, but it's going to get processed and spliced and capped and polyadenylated and all those things, and then it can go out and associate with the ribosome and make protein. Actually, we'll fold the protein. There we go. Okay. Is that does that address at all your question, or no? No? Are you not happy? I think Venus isn't happy. Okay, so we can talk about it, but that is kind of the big picture of what we're going to be learning about. Okay, any questions about what I just did? Okay. I keep hearing dinging. Who's dinging? Is that so distracting? I turned off all my notifications because I felt like I could never get something done. Then you're like, what? Might be Ed McMahon. Okay. Transcription in a nutshell. So this is the gist of it. All you need is RNA polymerase to bind to a promoter, and then it's going to open up. You'll have your transcription bubble. So your DNA is going to open up. And then once RNA polymerase clears the promoter, you get elongation of your transcript until you hit your terminator, which is the red part, and that's termination, and then you're done. that's pretty much the gist of it. It's simple as RNA polymerase being directed where it needs to work, and then making a little RNA copy of that gene. Okay, question? No? No. So I always like to start simple. Look at a little bit more detail. Okay, so this is what I said, process of gene expression and DNA, RNA, protein. Remember though, there's other RNAs being made. What kind of RNAs are being made? Is it all RNA that's going to result in protein? There's tRNAs. What else? What other RNAs? siRNA, mRNA, microRNA, in general, yep. What else? Huh? Ribosomal RNA. Yep. Then there's the, here I have a list coming up, but there's a lot of RNAs. Now, are there RNAs that are small RNAs that are not involved in? So, ribosomal RNA. What's the role of ribosomal RNA? What does it do? Is it the transcript for making ribosomes? Now, what role does it play? Yep. Right, so it's structural. Right. It's the skeletal I like that skeletal framework for the ribosome. So it provides a structural role. Does anyone know of any other biological molecules that use RNA not for the purpose of making protein, or not as a transcript for making protein, but for actually plays a role in a structure of something? Yep. Transfer RNA. It's not really structure. It does have some base pairing going on, but it plays a different role. It's not encoding a protein. It's bringing in your amino acids. But how about something that maybe more plays a structural role? Did anyone ever hear of SRP? SRP? Signal recognition particle. Spliceosomes are another one. Yeah, spliceosomes. They have snRNAs. Yep. I'm gonna call Dr. Snyder. I'm gonna get her down here right now. SRP is a molecule that recognizes signal sequences and brings proteins that are coming off the ribosome to the ER if they're destined for the circulatory pathway. And the structure of the SRP is pretty much dictated by RNA. Yeah, so RNA also plays a structural role. It's important to just acknowledge this and appreciate it. Okay, so genes can be translated, transcribed and translated with different efficiencies, right? You can have two different genes, and one gene might result in a ton of RNA, and another gene might only result in a few copies of RNA. Okay, why does this matter? Why? And this is on purpose. Sometimes you're going to get tons and tons of activity of RNA polymerase resulting in tons and tons of RNA. And sometimes very low levels of RNA. Why? What's the purpose of this? Why would we want this to happen?
It would be like to elicit a stronger response or a weaker kind of response.
Okay, so it's going to control the response. So if you're inducing gene expression in response to something, if you make a lot of RNA, you might have a stronger response. If you make a lower level of RNA, you might have a weaker response. What else? Does anyone want to add to that? It's a regulation mechanism, because if you have a lot of RNA, most likely will you have a lot or a little protein. And I say most likely because there's exceptions. All right. If you have only like a few copies of RNA, most likely will you have a lot or a little protein? A little, and controlling the amount of protein affects the response or whatever process the protein's involved in. For example, if you're making antibodies in response to an infection, you might make tons and tons and tons of RNA that are gonna encode those antibodies, right? They're gonna be translated into making antibodies because you want to secrete tons and tons and tons of antibodies so you can neutralize whatever virus is infecting you. Whereas something that's tightly regulated, that you only need a little bit of activity, I can't think of an example off the top of my head, you might want just a little bit. And you also might control the half-life of that protein. Who said something? Yeah.
Like clotting, clotting
Clotting factors. You don't want too much clotting factors. Yeah, that needs to be regulated. Is that regulated at the transcript level or at the protein level? Do you know? The protein level? So is that maybe, so there's two ways to regulate things. One, you can regulate how much RNA is made. Then you could also regulate how much protein is made and how long that protein stays around for. Yep. Okay. So let's talk about RNA for a minute. So there are three main structural differences between DNA and RNA. Okay, they're all right here, but who can think off the top of their head at differences between the molecules? Yeah. Ribose versus deoxyribose, right? It's right in the name. What else?
Base,
Use different bases, right? Which ones? Huh? Uracil is there instead of thymine for RNA.
We're just gonna say structural stability.
Structural
DNA is more stable than RNA.
DNA is more stable than RNA. Why?
Well, DNA. Reactual.
To what?
Double-stranded.
It's double-stranded.
And RNA is just one.
RNA is single-stranded. But is it truly?
Yeah, hairpins.
But is it truly single-stranded? Did you want to add something? Because it has that extra hydroxy. Um, Well, I don't know. It could be extra credit when a student answered it. Everyone copies it. I don't know. So tell me about it. I want you to expand on this. The react, because they're reactive.
I think it makes it polar.
Because that oxygen can go and attack things. Is that true? I don't know. Who agrees with that? Who feels better? I mean, yes, I know hydroxyls are reactive, but like, is that the reason why RNA is less stable? And the question is, see, I always think of it, and this is pure speculation, right? This is not science. Everyone turn off your science. We'll talk speculation for a minute. I always think the double-stranded nature of DNA makes it more stable. And that the single-strand nature of RNA makes it more accessible to nucleases. But due to the fact that RNA has secondary structure, right? Do you guys think RNA has secondary structure? Do you guys believe that when I say it? That should make it less accessible to nucleases. But then I guess then you can say structure of RNA is probably related to its stability. Oh God, there's a lot to think about here. You guys always get me. Yeah?
How many bonds uracil makes with adenine?
2.
2 bonds. Yeah, I was thinking, because if there's a difference between bonds, it makes one bond rather than two.
Oh, yeah, okay, yeah, yeah, no, no, so the question was about, base pairing and whether or not it's different, but no, it's pretty much the same. But yeah, so things to think about, I think I need to think about that a little more. Right, so sugars are different, ribose versus deoxyribose, yes.
I was gonna say RNAs are more mutations. Because once they're out, they don't have translations.
RNAs are more prone to mutation because they don't have a template for repair, you mean? But doesn't matter. But doesn't matter.
Sometimes it does. Because if you have a different base pair in an RNA, and you're trying to express a gene, it's gonna bring in a different codon. That's gonna affect the structure.
So now we're starting a very interesting conversation about the role, and I was going to get here later, but we may as well talk about it now, about mutation in RNA and does it matter. Okay? So mutation in DNA is not good because all of the RNA that you make will have maybe a different base, maybe a different amino acid, maybe will affect protein function, right? But in RNA, if you have a mutation that happens in the RNA, it doesn't matter. Yeah.
I guess it doesn't matter because you will have multiple other RNAs that aren't mutated and will express the correct proteins, and whatever protein you express could be refolded or encapsulated or
Okay, so that's one thing. So if you have mutation of one, let's say you make 10,000 copies of an RNA and one gets mutated because of some environmental toxin or something around, some drug or something. It doesn't matter because that one might not work well, but the other ones will be fine. So do you want to answer that?
Yeah, I was gonna say, it may not matter based on where that mutated, where that mutation in the mRNA or RNA is located. If it's on the, say, 3', the 5' or 3' end, it's not going to transfer. Or if it's an intron or part of the exon that might be removed, then you want to.
Okay, so you're saying if a mutation happens somewhere that's going to be processed away and not translated, it won't make a difference. Which you could say the same about DNA too, right? If it's in some kind of region that will never end up translated, right? Okay. Does anyone else want to add to this? Who thinks that mutation is a really big problem in RNA? I feel like you're really disposable because really your genome is the thing you really want to protect because that's your recipe, right? Without, if you mess up your recipe, your ZD will always be bad, right? Whereas if you burn, whereas if your copy of your recipe gets spilled on, you can go back, right? You can spill your ricotta cheese all over your copy. You can go back to your cookbook and always copy it out again. It'll be fine. Now, and you're not even going to try to make ziti from a copy of a recipe that has ricotta cheese smeared all over it because you can't really see it very well. It's going to try to make Okay. So, but I liked you bringing that up because we're about to talk about it anyways. Anyone want to add to this conversation? Yes.
What about telomeres ends? I mean, we have in DNA, but we have 48 tail, but is it strong enough compared to telomere ends in terms of protecting the DNA?
Okay. Telomere ends. So your telomeres.
Two telomeres.
Your telomeres. What's your question about telomeres?
I'm just trying to compare them to RNA poly-A tail, like in terms of being strong enough to protect your RNA versus DNA.
So I'm having a hard time relating them. Does anyone want to take a stab at this about relating poly-A tail in your RNA to your telomeres
DNA.
DNA and whether or not,
That's affect the stability of
whether or not, okay, whether or not, so your RNA and your DNA, your telomeres, your chromosomes are slightly unstable, but for a different reason, and than your RNA and your poly-A tail on your RNA protects the stability, because if you want to kill your RNA, remove your poly-A tail. Okay, and so you're trying to think about those two together and compare them, right? Okay, does anyone want to take a stab at this? Are there similarities, or where's the relationship? Yes.
I would say DNA would be more stable because telomeres would have some T within it, whereas poly-A tail wouldn't have that. So there are chances that it can get degraded easily compared to the telomeres.
Right, so your telomeres have the T-loop, which is a stabilizing structure. The other thing is so your telomeres are your ends of your chromosome, which are constantly shrinking, but where is that coming from? Like, what's the source of that? Well, telomerase fixes it, but why are they shrinking?
The
Primer removal and replication problem. So there's a source of that instability in a way, and then there's a built-in mechanism for fixing it. Whereas poly-A tail, yeah, it's just totally different. I'm having a hard time relating them. Does anyone want to add anything to that? I think RNAs are always much less stable.
So having that T loop, it's harder to kind of attack your DNA and rip it out. And, I mean, it's not really grab an RNA; it's just pulling it towards the cell. So and it's just hanging.
Right.
So you can like attack and just.
Right. So poly-A tail, exactly. By removing your poly-A tail, you can destabilize your RNA, and that's a mechanism for controlling RNA levels. So. Okay. Shall we move on? I like it. Everyone's thinking. Thinking is good. Right, so we've got this difference. You've got uracil and RNA. You've got thymine and DNA. That's the difference. What is this? DNA or RNA? RNA, good. Good, good. So DNA and RNA are different in structure. This came up also. Double helix in DNA versus a very specific secondary structure for RNA. Right? What determines the secondary structure of RNA? Okay, so hydrogen bonds, okay. And so like the base pair, you can have base pairing and between your different bases, and then you can have, you can also have within that secondary structure interactions. So here you've got your base pairing, and then here interactions between kind of different, so it's almost like an additional point of interaction. It's kind of similar to protein. Huh? So here are your bases. So you can have your bases base pairing, and that will form a secondary structure. And then you can also have bases interacting with faraway regions of your RNA. So when it all folds up, you kind of have what's similar to your tertiary structure that you see in protein. Right? So this is quite similar to protein, right? Where the interactions between your amino acids are going to form a secondary structure and kind of a tertiary structure. Okay? Yeah. No. Nope. I don't know. Oh, that's so sweet.
Well, I've heard proteins have both structures and secondary removal. What exactly is RNA's secondary structure and tertiary structure?
So you're having the structure which is the base pairing kind of structure between the, well, just adjacent or close bases. So that's forming these kind of stem loop structures. But then, so then within this, you're also gonna get the interaction between far away regions, which I'm calling tertiary, which might be wrong.
But why would it have to get folded and make
It's spontaneous. So, why would RNA have to get folded to make secondary structures? In some cases, it's for its function, right? In some cases, like tRNA has a very specific function, very specific structure. The RNA that's involved in making ribosomes or making SRP, those have specific structures and functions, right? But they're spontaneous because they're dictated by their sequence.
So they are not always open when they're being translated.
They're not always open when they are being translated. Well, they have to open so they can go through the ribosome. Yep, but there are secondary structures, and then the structure of the RNA dictates also whether it can be translated, because there are sometimes stumbling structures. It dictates, it's a form of regulation also. Yeah. Hydrophobic versus hydrophilic regions, because so there's a lot more diversity when you're looking at amino acids, right, than when you're looking at bases. So I think it's a lot of hydrogen bonding. Huh?
I was just wondering if there's anything related between that.
Yeah, I don't see that. But maybe I have to think a little deeper, but I don't think so. Everyone good? Okay, so RNA polymerization occurs 5' to 3', similar to DNA. Nucleotides are added in the order of DNA sequence. Everything you know, right? So you have your DNA, and one strand will serve as your template strand. It doesn't matter which strand. And what really matters is the orientation in which RNA polymerase is recruited in the end. So one strand will serve as your template strand, and then you'll get transcription, and then your RNA is going to be similar to your other strand in sequence, right? Except for your T's and D's. Yeah.
I'm confused on that part. So, how do you, not how, do you have to open your DNA again for that to happen? So is that done also by helicase?
Now, so the question is, do you need to open your DNA for this to happen? Yes, because you have to get in there to polymerize to make your RNA, and because it's going to be base pairing with your template strand. That's how it knows which bases to incorporate. But there is no helicase.
So what's open with the DNA?
The RNA polymerase, but now well, so chromatin remodeling complexes are opening up your DNA in the sense, in the sense that it's not wrapped around histones, but not physically opening the two strands of DNA, no. And then this is helicase independent, at least that's what I teach you. The question is what's opening it. We have to think about that. I'm sure I can. We can make it a stump the teacher, because that's when I should really be able to tell you. Right. It's a stump the teacher kind of day. So I just got asked whether helicase is required to open up the DNA for transcription to happen, and I said no, because that's what I teach. But then Tammy asked, well, how does it open? And I'm like, RNA polymerase does it, but that was hand waving. So we need more detail. So if you can find me the detail of exactly how.
Isn't the transcriptional all of them?
I want to know exactly what acts like a helicase to open up the DNA, because it doesn't spontaneously open, does it? Something must be opening it. And I need it to be like like a real, don't copy paste, don't Wikipedia. Wikipedia is nicely referenced, go to the reference, yeah.
What access do they still for transcription?
What acts like a helicase to open DNA during transcription? Oh my god, I know!
It's too late. We have some recording.
Are you gonna give us?
I'll let you try, but I'm embarrassed. It's okay. You can't always be on your toes. Try. It's okay. You guys, you know what? I know. I know, and you guys have, it's gotta be in one word.
That's not right.
Because it's one thing. Yet it's one thing that does it. Oh! I'm so mad right now. And everyone who's listening at home, you're probably giggling. But you guys, one word. I don't want some long, blah explanation because it's one thing that does this. One word.
So we don't even need to cite it.
You do not even need to cite it because there's nothing that even needs to be cited because it's
In the book.
it's oh yeah, it's in the book already. You guys get me because my brain is still a little bit on the cell cycle. That's the problem. Okay. Okay, so we're talking about kind of compare and contrast here of transcription and DNA replication because they both involve an enzyme that's involved with opening up DNA, and then bases are brought in, and you're polymerizing nucleotides, right? So there's some similar things. Like for example, the sequence is determined by base pairing, just like synthesis. And then synthesis occurs 5' to 3', right? 3' OH. Similar. But then there are differences. So if you look at this, so this is showing, here's your DNA, that's the yellow helix. And this is showing your RNA polymerase in the process of transcription. And in the blue here, you can see your RNA kind of coming out the end, like comes out behind. Looks like, it's like poop. Sorry, how'd you do it? I always look at it and I'm like, it's like a fish, and is the fish pooping? Okay. Now you guys, have you never seen a fish poop? My dad had
Why goldfish?
you know. I had to
so you can write it on our exam.
No. It's the RNA exit channel. Okay, anyways. Sorry guys, totally. I'm having an unprofessional week in general. Okay. Anywho, there's base pairing going on, right? So here you can see your, this is your DNA RNA helix right here. There's this very short region going on, and that's because it's the base pairing that's determining which base is being brought in and added to the growing RNA. And then as the enzyme moves to your right, in the direction of the arrow, this is moving out, the new RNA is coming out to the RNA exit channel, and new nucleotides are coming in and being added. So what's different if the RNA is displaced, right? Once you add your bases, it's being displaced, meaning it's being kicked out. It's not stuck base paired to your DNA, right? When you do DNA replication, your DNA is base paired, and that's it, right? It's never displaced. But here, your RNA is displaced from the enzyme and goes a different way. And then, of course, the RNA that's produced from transcription is much shorter than the DNA produced from replication, right? You're just doing one, you're just transcribing one gene. when you're doing transcription, whereas DNA replication is replicating your whole genome. That's very long DNA being produced. Okay. Any questions about this? Yes.
I know that the RNA polymerase doesn't have exonuclease activity like the DNA polymerase, but we.
Well, it can back up.
Inside the
It can back up and cleave. Yeah. It's not. What's it called? Yeah.
Okay, so I was thinking that, like, you know how in the DNA polymerase, how it moved down to like that exonuclease site?
It's
It does
different.
the same thing.
This is different.
Okay.
Yep, so exactly. So compare and contrast proofreading capabilities of your enzyme. DNA polymerase and RNA polymerase, those have a different structure. This doesn't really look like a hand. We're going to call it a fish. Good thing said. I know, completely unprofessional. You can get me fired. But what it does, so with hand, yes, it's got this exonuclease domain that if a base is added incorrectly, the DNA moves there, gets cleaved off, and then it goes back and continues. Here, what it does is it just backs up, cleaves off in the same place. And so it's not like it moves into a different domain. It just backs up, removes the incorrectly added base, and then keeps going. And we'll get to that a little more in a minute. Hopefully. Yes,
I don't know if this is a dumb question, but are the
I just talked about fish food.
are the base pairs just like flooding in and whatever fits? It's not getting like specifically chosen to get its
Right, it's not like, so are you thinking
So, like-
about ribosomes where like there's like more,
I
well
guess.
no, I mean, okay, so your question is about
How does it get
how the right bases get to where they go. So you can imagine in your nucleus, there's a high concentration of nucleotides, right? And what's going to be added is going to depend on what base pairs with the DNA template. That's it.
Okay.
Yeah.
Isn't it basically just what has the stronger association to
Right, whatever binds most strongly to the base pair is going to be there long enough for, I believe it's pretty much the same kind of nucleophilic attack mechanism where, because you're still adding to your 3' OH, you still have a triphosphate. That's all the same. There's still magnesium in there to kind of hold everything in the right place. Did I teach that? Anyways, so really, it just needs to interact strongly enough so that the reaction can happen.
Okay.
You can imagine if it's not going to bind strongly, it won't be there long enough.
Yeah.
There's probably even a better answer than that, but that's the way I see it. Anyone else? Right? Okay. So there are many RNA polymerases transcribing a gene at the same time. Did you guys know that? It's not just one. So like we're always taught that it's like this, and it's just like this RNA polymerase going along, making an RNA, but it's actually more like this. Okay, this is actually showing two different genes, and then each line. So this is the DNA, and then each line coming off is another transcript being made. So you can imagine, I don't know, there's like probably thousands being made of each gene there. And then I believe, I'll have to check the credits, not the credits, but the figure legend, but I believe this is already ribosomes associated. This is in both. But, so lots, it's like pretty much an RNA polymerase is going to hop on and start transcribing, and another one's going to hop on right behind it and start transcribing. So there's this successive transcription of multiple transcripts at a time. So the RNA polymerase moves pretty fast. The rate of nucleotide addition is about 20 nucleotides per second. Question? You're confused.
no, what are you saying? Is it all of the leaf-like things, or RNA, or is just that one strand in the back there?
So let's use this. So this right here is your DNA. That's DNA. That's one gene, right? And then this over here is another gene. Okay? And so each little thing coming off is an RNA. So you can imagine all these black dots here are RNA polymerases on here. And then these are the RNAs coming out the RNA exit channel.
This is eukaryotic, right? Not prokaryotic.
This is prokaryotic, I believe, because of because I think those are ribosomes attached, and that when in prokaryotic cells that could happen, but in eukaryotic cells you have to have all the processing and then. Yeah. But we can check the figure logic. Okay. So yeah, so nucleotides, about 20 nucleotides are added per second. Is that fast? I think that's pretty fast. So you can get over 1,000 transcripts from one gene in an hour. And it all, of course, depends on the size of the gene and all that. This is always one of those mind-blowing pictures for students. Whose mind is blown and who's like, I've seen this before. Mind blown? Okay. Okay, so we've got some differences still between RNA polymerase and DNA polymerase. So the structure is completely different. Like I said before, there's no hand for RNA polymerase. Your bases are linked by a different sugar, right? Deoxyribose versus ribose. And so there's a different enzyme activity associated with that. No primer required. Right, DNA polymerase, you need a primer because DNA cannot be synthesized from nothing. But RNA fine. And then it's not as accurate as DNA polymerase, but it does have proofreading. So it can back up and excise the incorrect base, like we just said. So the RNA polymerase error rate is about 1 in 1,000. That was about what we thought for HIV's reverse transcriptase, right? Something like that. That's a much higher error rate than for replicative DNA polymerase. And so we just had this conversation about why this higher error rate is tolerated, so we don't need to have it again. But pretty much, if you make a mistake with one RNA, who cares if you're making over 1,000 copies of it, as long as most of them come out okay. Because you can imagine if it can't make a good protein, then it's never going to affect anything. It's going to kind of just be degraded anyways. Yes?
I know this is probably different in different RNAs, or RNA calls and DNA calls, but in general, which synthesize
Which
faster?
is faster? Oh, you know what? We can do that math. Which is faster, RNA polymerase or DNA polymerase?
It's less.
Really care about the states.
I guess if there's less information,
Okay, so what I'm hearing is there's less slowing down to fix. I don't know. There's a higher error rate, so you can guess that. I don't know, actually, because then you have your intrinsic enzymatic activity. So in the textbook, it does say, just like it says 20 nucleotides per second, we can easily look up how many nucleotides per second DNA polymerase or can add. But I'm not 100% sure. I would think DNA polymerase, though, because of its large job it has to do, but I don't know. Okay, these are different types of RNAs, right? We went through a bunch of them. You got your mRNAs, ribosomal RNAs, remember again, structure of the ribosome, tRNAs, small nuclear RNAs, that's your splicing, right? Your SNO RNAs, processing ribosome RNAs, your small cagal RNAs that modify other RNAs. microRNAs, which you guys should know all about because of this week's recitation paper. By the way, how's that going for people? How's it going getting through that? Are you meeting with friends to do it, or are you doing it on your own mostly? Friends? Okay, I've had some people tell me they're having a hard time, so I want everyone to kind of reach out to each other and try to get through together because we do like why suffer alone. siRNAs, those also are involved in turning off gene expression. And then there's other non-coding RNAs, such as the one that binds to telomerase, right? That template telomerase hold has to come from somewhere. Inactivation, transport of proteins to ER, right, the SRP. So lots of RNA produced. Yep.
So mRNA could lower the expression of a gene, but siRNA will turn it off.
Okay, what is mRNA? Are you saying microRNA?
Yeah.
miRNA?
Yeah.
miRNA and siRNA are the same function. Both of them regulate gene expression, often by interfering with mRNA stability or translation. The difference between microRNAs and siRNAs are structure. MicroRNAs tend to be hairpin shaped that are then processed, whereas siRNAs are small double-strand RNAs. They both end up, well, I would overlap them in many ways in structure and function. Yeah.
RNA editor that was copying like one sequence into like making two different proteins.
Oh, are you talking about RNA editing?
Yeah.
Okay, what about?
Because that's falls in this category.
No, so RNA editing, those are proteins that will change, often will change bases in RNA to change, maybe to put in an early stop codon that will result in two proteins, like the cytidine deaminase that we talked about, where it was the apolipoprotein B. In one case, it's a long protein that does one thing, and in another case, it makes a premature stop codon, making it shorter, or like antibodies secreted versus membrane bound. Yeah. What's the purpose of double-stranded RNA? Now, do you mean in terms of
Why would it be double-stranded? Why?
in terms of like why would you let it? Why would base pair and do things?
I guess it was just not a concept that was much.
Right, because you originally just taught RNA single-stranded. Here it is. Why would a base pair? I mean, it's just that's a normal function of a sequence, right? It's just what it does. I think you will never find, complementary. You'll never find just linear RNA sitting. I think it's always going to want to hold up on it. Yeah.
So it's not always two different RNAs. It could be
No, it's usually one thing that's kind of stumbling structures. Yeah. Okay, you guys go up.
2H.